I now call to order the lecture portion of the Society's 2524th meeting in what is now the 154th year since it was founded on March 13th, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., dedicated to providing a forum to further scientific understanding and inquiry to as broad an audience as is interested. We welcome our members, guests, and friends to the lecture part of tonight's PSW meeting and the lecture by John Sunderland on medical imaging with antimatter, high-resolution positron emission tomography. Let's begin by thanking some sponsors. The Society is grateful to PSW Full Year Series sponsors, PSW member Mike Helton and Helton Associates, LLC. Thank you, Mike. And to PSW member Joe Schulman and the Schulman Foundation. Thank you, Joe. And the sponsor of tonight's lecture, PSW, Michael Sandifer. Thank you, Michael. You should know that the major sponsors account for about 35% of our revenue, and the lecture sponsors account for another 20 to 30%. So they are essential to us remaining solvent. And so anybody who wants to become a major donor or wants to sponsor a lecture, please see me. We are now filling in the lecture sponsorships for the rest of this fiscal year, which runs from June 30th. I say that right? July 1st through June 30th. Everybody raise your hand. No, I'm just kidding. I'd like to tell you something really nice. We have a matching grant, 2-for-1, up to $10,000 of matching, so $30,000 in all. So this is an auspicious time for you to make a donation. It runs from November 1st through the end of January, so you can decide which tax year you want to make your donation in. And with that, let's just say I am pleased to announce the following new members, some of whom are in the audience, and we welcome them. Millie Ho, process development scientist with AstraZeneca, interested in physical and biological sciences and medicine, who learned of PSW from PSW member Will Angel. I think. That's what you said. Judah Rosner, a molecular biologist with NIH interested in quantum physics and infectious disease, who also learned of PSW from a friend. Stephen Feinstone, a physician scientist interested in infectious diseases, virology, vaccines, and modern physics and cosmology, who learned of PSW from PSW member and lecture sponsor, Michael Sandifer. Antoinette Greenberg, a nurse educator with ACG coaching and Chicago family tutor, interested in the history of mathematics and science and the transmission of scientific thought, including archaeology and gene mapping studies of medieval England, who learned of PSW from PSW's videos on YouTube. Joseph Crandall, a software engineer with Microsoft, interested in quantum computing, who learned of PSW from friends. Stephen Schlashinsky, an aerospace engineer with SAIC, interested in physics, astronomy, and satellite astronomy technology, who learned of PSW on the web. And John Sunderland, tonight's speaker, who has he hasn't, whose interest will be clear in part from his lecture tonight. So please join me in welcoming them all to membership. All members are entitled to a signed copy of Compendium Volume 17 of the Bulletin of the PSW. If you've not received yours, please come see me after the meeting. It's notable that all members are invited to the pre-lecture receptions and dinners with the speakers for camaraderie and interesting discussion. So if you are interested in joining, take that into account. If you're interested in becoming a member, you can access a membership application on the PSW website by clicking the blue Join button on the upper right-hand corner of the homepage. And if you're here in the Powell Auditorium, you can access the application using the QR code on one of the business cards set out on the tables at the back of the room. Don't be shy. Please go ahead and use your phone and the QR code. Everyone with a genuine interest in science is welcome to apply for membership. Recording Secretary Scott Matthews will now present the minutes of the 2523rd meeting and the lecture by Neil Johnson on the science of when good AI will go bad.
Thank you, Larry. Good evening. On October 17th, 2025, members of the Society and guests joined the speaker for a reception and dinner at 5:45 p.m. in the members' dining room at the Cosmos Club. Thereafter, they joined other attendees in the Powell Auditorium for the lecture proceedings. In the Powell Auditorium of the Cosmos Club in Washington, D.C., President Larry Milstein called the lecture portion of the 2523rd meeting of the Society to order at 8:10 p.m. Eastern Time. He began by welcoming attendees, thanking sponsors for their support, announcing new members, and inviting guests to join the Society. Scott Matthews then read the minutes of the previous meeting, which included the lecture by Adam Rees titled Measuring the Universe, From Hubble's Discovery to the Hubble Tension. The minutes were approved as read. President Milstein then introduced the speaker for the evening, Neil Johnson of George Washington University. His lecture was titled AI's Jekyll and Hyde Tipping Point, The Science of When Good AI Will Go Bad. The speaker began by describing the layers in generative AI in analogy with people in a movie theater, passing messages forward from one row to the next. He described how the series of layers creates the next token based on its training and appends that token to the prompt and cycles it back to the input layer to repeat the process. He used the term attention head to describe each of the computational units making up a layer. He said that even the people writing the code and defining the structure of large language models don't actually understand how they work. He said that this lack of understanding leads to a lack of trust. He gave the example of flying in a modern jet airplane, saying that even if we do not personally understand the physics and engineering of how the plane flies, we know that someone does, and that therefore we have trust that the airplane will get us to our destination. Johnson described some of the recent headlines about the way generative AI is being used, including personal relationships, government reports, driverless cars, emotional therapy, medical diagnosis, and plumbing. He said that the ramifications of generative AI producing bad output is serious and is, quote, not like getting a bad glass of beer, unquote. The speaker discussed the idea of an embedding space, a multidimensional space where data like words, images, or other items are represented as vectors. He discussed the fact that an AI's training would group similar tokens in similar locations in the embedding space. He said that the input or prompt would send the first layer of the AI to an initial point in the embedding space, and that subsequent layers would generate additional tokens appended to the prompt. He described how each additional token constitutes a displacement vector, ideally moving the AI closer to a, quote, good answer. He described the evaluation of each displacement using the dot product. The speaker discussed how this iterative process can create bad tokens or incorrect output by giving the example of asking, quote, what's nice in DC, end quote. If the dot product between the tokens for nice and trees is very large, the displacement vector of the subsequent token can move the AI's position in the embedding space from DC into Maryland because Maryland has more trees. He mentioned techniques that AI companies use to prevent, quote, undesirable output. These included fine-tuning and retrieval-augmented generation. Retrieval augmented generation works by first retrieving relevant information from an external knowledge base or document store, and then using that retrieved information to augment the LLM's response before it generates the final answer. Johnson claimed that recent research indicates that the inclusion of such augmentation may create instability in the output of AIs. The speaker presented a formula based on the dot product for calculating the, quote, tipping point, the point where an AI begins to move towards a bad or harmful output. He discussed how changing the prompt submitted to an AI can change its tipping point, including using polite prompts like the word please. He claimed that using his formula, we can now have a more informed discussion about prompt engineering. He presented a two-dimensional representation of the embedding locations of 3 words on ChatGPT: Earth, flat, and round. He claimed that the embedding distance between Earth and flat was very close to the embedding distance between Earth and round, leading to the possibility of the AI generating an incorrect result for some prompts about the shape of the Earth. He explained that the proximity of these 3 words in embedding space is due to the fact that the AI is trained on social media, and that there are many people on social media who believe that the Earth is flat. The speaker ended his talk by commenting on future potential issues with AI. These included problems with scaling as AI becomes much larger, stochastic algorithms generating random results, and agentic AI, systems that can autonomously plan and execute actions with limited human intervention. The lecture was followed by a question and answer session. A member asked about the similarities between electronic neural networks and the neural network of the human brain. Johnson responded, quote, structurally zero, no connection at all, end quote. He said that functionally they are quite similar, particularly in terms of the attention mechanism. He said that both AIs and the human brain have limited attention, so that attention on one particular idea or input comes at the expense of others. A member asked about the ultimate limits on scaling of AIs. If we had unlimited electrical power and significant economic drivers, how big could they get? Johnson responded, quote, I don't know. I don't think anyone knows, end quote. He said he could see no reason why it would not continue to grow at an increasing pace. A member on the live stream asked how you set the values for the vectors G and B, good and bad. Johnson responded, the challenge is determining what's good and bad, end quote. He said that for questions about the shape of the Earth, we know what good and bad answers are. He said that for something like tax compliance, a priori knowledge of good and bad is easily determined. For mental health advice, it is not so clear. After the question and answer period, President Milstein thanked the speaker and presented him with a PSW rosette, a signed copy of the announcement of his talk, and a signed copy of Volume 17 of the PSW Bulletin. He then announced speakers of upcoming lectures and made a number of housekeeping announcements. He adjourned the 2,523rd meeting of the Society at 9:58 p.m. Eastern Time. Temperature in Washington, D.C., 12.8 degrees Celsius. Weather, fair. Audience in the Powell Auditorium, 112. Viewers on the live stream, 26, for a total of 138 live viewers. Views of the video in the first 2 weeks, 309. Respectfully submitted, Scott Matthews, Recording Secretary.
Thank you, Scott. Excellent job, as usual. Are there any comments or corrections? Hearing none, I will entertain a motion from a member to accept the minutes as read. I have a motion to have a second. I have a second. All members in favor? All members opposed? All right. Minutes are accepted unanimously as read. They will be posted to the website in due course, provided there's nothing that comes in over the web that requires a change. And we now turn to tonight's lecture by John Sunderland. John is professor of radiology and of physics and astronomy at the University of Iowa. where he also directs the Positron Emission Tomography Imaging Center and the Small Animal Imaging Core Laboratory. He is widely recognized for pioneering efforts that expanded clinical PET imaging, in particular through centralized radiopharmaceutical production and distribution. And he was the principal author of the University of Iowa's new drug application for gallium-68 edotrietide. Probably got that wrong, but you can correct it when you're up here. And he has been a leading figure in advancing PET internationally, particularly PET standardization. His recent work is focused on improving the accuracy and reproducibility of quantitative imaging across different scanners and clinical sites, and he currently leads a global collaboration developing quantitative SPECT imaging standards to support radiopharmaceutical therapy trials. He's an author on more than 250 scientific publications. Among other honors and awards, he's a fellow of the Society of Nuclear Medicine and Molecular Imaging. He earned a BA in physics at Williams College, an MS and a PhD in medical physics at the University of Wisconsin-Madison, and an MBA at Centenary College, Frost School of Business. All questions will be fielded after the lecture during our usual question and answer session. John, the stage is yours.
Okay, well, thank you, Larry, so much for the kind introduction and the invitation to be here. I love talking about this stuff. I've been doing it for 40 years now, and it is just about the most interesting thing I could ever imagine doing. Positron emission tomography, at its essence, is multidisciplinary. It requires an understanding of physics and biochemistry and synthetic organic chemistry and medicine and engineering, all that are involved in making this work. And we're going to cover all of this a little bit, so regardless of what your background, something is going to probably be kind of in your wheelhouse. So as advertised, we're talking about medical imaging with antimatter. It's about positron emission tomography. The way I'm going to start this is with an example, and then we're going to get down to the details. So first and foremost, PET doesn't image anatomy. PET images the specific biochemistries in the body. Biochemistry that is visualized is absolutely dependent upon the radioactive drug that we inject into the patient. In this patient here, we're imaging glucose metabolism. So how are we imaging glucose metabolism? We see glucose metabolism because we've injected a tiny, tiny amount of radioactive glucose into this patient's body. That brings up a couple of questions. One is, what's a radioactive glucose analog? Secondly, what does this have to do with antimatter? Third, how does this ultimately manifest as a medical image? And lastly, why do we even care that we can actually do this, as amazing as it actually is? So we're going to answer these quickly one at a time. What do I mean by a radioactive glucose analog? And just so you can kind of know what's going on, about 80% of the clinical PET scans that are done with this molecule. Very common. But there are a number of others, and we'll talk about those kind of as we go on. So what is a radioactive glucose analog? This is glucose. Glucose is a 6-member ring. It has a bunch of oxygen and hydrogens kind of hanging off of it, like you see here. That's glucose. This is what was injected into the patient. It's 2-fluoro-2-deoxy-D-glucose. The only real difference here, the only actual difference, is in the 2 position that OH group, sitting right there, has been replaced by a fluorine atom. Otherwise, it's identical. F18 is radioactive. It has a half-life of about 2 hours. And it behaves almost exactly like glucose in the body from a biochemical standpoint, at least in the beginning of its metabolism. Okay, now what does this have to do with antimatter? Well, it turns out an F18 nucleus is fundamentally unstable. There's an imbalance between the number of protons and neutrons that are in there. There's too much positive charge. It wants to somehow get rid of that positive charge. And so the way it does that is by jettisoning a positron. A positron is an antimatter electron. What ends up happening is a proton in the nucleus turns into a neutron, and that positron comes meandering out. It goes only about 0.2, 0.3 millimeters before it comes to rest. And when it comes to rest, it will interact with one of the many ubiquitous electrons that are lying around, and they'll annihilate. Actually, before it annihilates, it'll form positronium for about 10 to the minus 18th seconds. They'll kind of orbit around one another, and then they annihilate. They disappear. An electron has a mass of, 9.1 times 10 to the minus 31st kilograms. So does a positron, but when they annihilate, that mass goes away. In its place, you get energy. But it's not just some amorphous, ambiguous energy. It's two 511 keV photons that go off in exactly opposite directions. They go off in opposite directions because functionally the positron and the electron were at rest to begin with, so the momentum of the system beforehand was zero. So the momentum of the system afterwards has to be zero. And that's just that whole E equals mc squared. That's where the 511 keV energy comes from. Those are the rest mass of the positron and the electron. So how does this manifest as a medical image? Well, the way we do this is we've injected that radioactive glucose into the body, and this is just one of those fluorines on one of those glucoses will decay by positron decay. It'll emit two 511 keV photons going in opposite directions. There are tens of thousands of tiny scintillation crystals around the patient, and when two of those detectors detect those photons at exactly the same time, then, and exactly the same time means within about 5 billionths of a second, then you know that the event happened somewhere along that line connecting those two detectors because those photons go off at 180 degrees. We don't know where along that line, but we know somewhere along that line. We detect tens of millions of those events during the course of a scan, which takes between 10 and 20 minutes if you're trying to do kind of a whole body scan. And then all that projection data, those tens of millions of events, are mathematically reconstructed to generate the most likely distribution of radioactivity in the body, and it'll create the image here. And we're going to go through all of those steps in a couple minutes. And now the question is, why do we care that we can do this? We care because we understand a lot about the human body. We care because in cancer biology, we know that cancer cells almost always metabolize glucose really inefficiently, using the aerobic glycolytic pathway where you only get 2 ATPs when glucose is burned. Normally you get 38 or something when it's burned. It's very inefficient. So cancer cells need to burn glucose like crazy in order to support cellular function. So when we inject this radioactive glucose, it goes in the body where your body uses it. You can actually see a lot of activity in the brain because the brain uses glucose, right? But it turns out cancer cells use glucose too, which is what you see here. This is a patient with breast cancer. They thought it was metastasizing. They didn't know where. The CT scan came back normal. So they did a PET scan. And these are 4 lymph nodes that have cancer in them where the cells are dividing very rapidly, taking up glucose very avidly. And so we see it here. So, want to point out here that PET scanning always is a tracer methodology. When I said we only inject a tiny, tiny amount of radioactive glucose, I really mean it. About a millionth of a single grain of sugar is all we're injecting into the patient. It's dissolved in saline, but that's the mass quantity of the drug. We are not perturbing the system we're studying. We never want to do that. There's zero pharmacological effect to the patient when in PET scans because we're always below any kind of remote pharmacological dose. Despite that, the FDA considers this a drug, and so we have to manufacture it according to good manufacturing practices, aseptic processing, the whole 9 yards. But physiologically and pharmacologically, there's really nothing going on. So I want to point out that this fluorodeoxyglucose that I mentioned, we didn't just kind of develop it willy-nilly. We did it with a firm understanding of what we were doing. So this is glucose. I could label glucose with carbon-11, which is a positron-emitting isotope of carbon, right? So I could do that, and it could go through its metabolic pathway, and it would go on through, and ultimately it would be completely metabolized. We end up with 6 water molecules and 6 carbon dioxide molecules and a bunch of ATP. The problem with labeling this with C11 is because if you inject it in the body, it gets metabolized in the cells, and it starts turning into carbon dioxide. What's the carbon dioxide going to do? It's going to start migrating out of the cells, into the tissue, into the blood, into the lungs. First of all, you're going to start breathing it out, which is a problem in and of itself. But mostly, we're imaging the C11. And if the C11 isn't staying sequestered in the cells like glucose and it's kind of all over the place, we end up with this muddy, blurry picture of where the C11 is and not where the glucose metabolism is going on. So what we do is we made this FDG, this fluorodeoxyglucose, with the F18 in the 2 position. It gets transported in the cell just like normal. A normal glucose does through facilitated transport. Hexokinase phosphorylates it. By the way, FDG 6-phosphate or glucose 6-phosphate can't leave the cell anymore. It's gone in, it can't go out. But then that next enzyme tries to act in the 1 and 2 position, and it's looking for that OH that's supposed to be there, but it's not. And so it can't metabolize any further. And so it gets stuck in the cell. And as more and more glucose is being metabolized, more and more glucose is being sequestered in the cell, and it's brighter and brighter and brighter. So the image ends up being functionally proportional to glucose metabolism. And we did this absolutely on purpose. So a lot of effort goes into designing these molecules to behave exactly the way we like them to. Okay, so that's the end of the example. So now I want to start talking about the rest of the talk in the organization. So first, we're just going to do some really basic radioactivity and really basic nuclear physics stuff, just so we understand what's going on. Then we're going to talk about cyclotrons and the production of positron-emitting radionuclides. We're going to move on to the diversity of PET radiopharmaceuticals that are out there, because it's not just this FDG glucose. There's thousands that have been studied over the last 40 or 50 years. Then we're going to talk about how PET scanners work, talk a little bit about how these images are reconstructed in a very general way, and talk a little bit about time of flight, because that's the direction the technology is going, and it's making our images and quantitation just all that much better. Then talk about some new technological developments, kind of novel PET scanners, but they're allowing us to do incredible things that we weren't able to do before. And then finally, it's the reason I'm here today, actually in person, because I was actually giving a talk up in Bethesda just yesterday on how the development of PET radiopharmaceuticals is leading to a revolution in cancer treatment. I mean, this radiopharmaceutical therapy stuff, it is so big, we're even being advertised on NFL games now. So we've hit the big time, finally, after being in the shadows for forever. So part 1, let's talk about the basics of radioactivity. Nothing hard here. But we're going to start with the periodic table, which to a nuclear medicine physicist is kind of a shallow 2D representation of a much more interesting 3D space. And what I mean by that is if we look at, for example, carbon, which is here, which is number 6, it turns out there's a number of isotopes of carbon. So there's actually kind of a bunch of things stacked up. Carbon-12 is mostly what we have in our body, 6 protons, 6 neutrons. We have some carbon-13 in our body. That's 6 protons and 7 neutrons. It's less than 1% of all carbon, but it's stable. But if you get an extra proton in there, and now you've got 8 neutrons and 6 protons, all of a sudden the protons and neutrons are in imbalance. It needs to get back into balance, and so a neutron decays into a proton by emitting a beta particle, and C14 has a half-life of 5,700 years. This is what we do carbon dating with. You can also go the other way. This is 6 protons and 5 neutrons. That's the carbon-12 that we could have labeled the glucose with if we wanted to. That has too many protons in the nucleus. It wants to get rid of that extra positive charge. It decays by positron decay. So each of these elements here has a whole bunch of radioactive and non-radioactive isotopes associated with it. So we don't look at the periodic table. We look at the chart of the nuclides. And so if you look at the very bottom here, so it's number of protons are here on the y-axis and neutrons on the x. So if you look very much down the corner there, that's hydrogen. 1 proton, 0 neutrons. That's the black dot there. Right next to it is deuterium. That's also black. Black means stable. That's deuterium. As you go up, it goes to hydrogen, and then lithium, beryllium, and so on and so forth. The black ones are all the stable ones. That's what we're all made of. That's what the world's made of. But there's a rich bunch of stuff, which is sitting here in the pink, that are radioactive. They are proton rich. And so these guys all pretty much decay by positron decay. So there's a lot of these that decay by positron decay. The ones down at the bottom that are in blue, neutron rich, they decay by negative beta decay. The yellow ones decay by, they're heavy nuclei, large nuclei, they generally decay by alpha decay. I didn't used to care much about the beta and alphas, but I care very deeply about those now because they're involved in the therapy part. But we'll get to that at the tail end. Okay, this is just a diagram of positron decay. You have a nucleus, which you see here, and what ends up happening is a proton, which is circled in yellow. That's going to turn into a neutron by virtue of emitting a positive beta particle, a positron, an antimatter electron comes out. An antineutrino also comes out. The two of them share the energy. Sometimes the positron gets most of the energy, sometimes the neutrino, sometimes split evenly. So there's actually a spectrum of energies there. And as we mentioned, that's not the end of the story. That positron travels a short distance, usually less than a millimeter. It annihilates with an electron. You get those 2 511 Ke photons. So that's what happens to all those pink guys on top. Negative beta decay is when you have too many neutrons, and so a neutron, which you can kind of see a blue one kind of circled in yellow, that ends up turning into a proton by virtue of emitting a beta particle, which is just an electron of nuclear origin. It gets emitted with a neutrino. They too share their energy, but nothing interesting happens to the beta particle. And with the large nuclei, when you have alpha decay, it emits this alpha particle, which is 2 protons and 2 neutrons. This is a lot heavier and has a lot more energy associated with it. Once again, I didn't care about this very much, but on this whole therapeutic side, alphas are becoming really important. And I'd be remiss if I didn't mention that when these nuclei decay, from one element into another by emission of a particle, they will often end up in an excited state. Just like electrons can be in an excited state, and then when they go back to the ground state, they emit a photon. Same thing happens in the nucleus. When a nucleus is in an excited state and it goes back down to the ground state, then it emits an electromagnetic radiation in the form of a gamma ray. That's what gamma rays are. They are photons of nuclear origin. X-rays are high-energy photons. They're of electron origin. You can actually have X-rays that are higher energy than gamma rays. It really has everything to do with where they come from. I have over on the right-hand side the energy diagram for lutetium-177. You don't really have to read it, except that this functionally shows that when it decays by beta decay, 80% of the time it goes down to the ground state, and there are no gammas. But that other 20% of the time it decays to an excited state, and we get gamma rays. I like gamma rays. Gamma rays are energetic enough they'll leave the body, and we can image them. We can't image the positrons. They only travel like less than a millimeter, not the beta particles. Alpha particles travel like 20 microns. We can't detect those. Gammas, we can detect. Okay, going to part 2. Cyclotrons and the production of positron-emitting radionuclides. So to do positron emission tomography, we need proton-rich nuclei. And these things I mentioned, half-lives of 2 hours, 20 minutes, right? You got to do this stuff, kind of on site. So how do you make a positron emitter? Well, the obvious way to do it is to take a proton and shove it into an existing stable nucleus, right? ChatGPT did this for me. They never do good diagrams. They did a good diagram here. But it's not as easy as that. The way we have to do this is to use a particle accelerator or a cyclotron. It's the fact that making positron emitters required cyclotrons that delayed the introduction of PET into the medical space. It was introduced about the same time as MRI. MRI took off right away. We had to build infrastructure around every major city when CMS, when Medicare wasn't reimbursing for these things. So it took a long time in order to get past the few universities that were actually doing this, demonstrating how important and amazing this technology was, until it finally kind of hit the mainstream. And the availability of high current, medium energy cyclotrons at a reasonably affordable price that were easy to run was a big step there. So that's what the medical cyclotron does. And this is the whole thing, right? So you have a stable target nucleus, which you see here. So that's just on the outside of the cyclotron. And the proton comes from the cyclotron. It ends up being accelerated towards that nucleus. Now, the reality, and so let's pretend this is an oxygen 18 target atom. That's what we use to make F18. Oxygen 18 is a stable isotope of oxygen. We have to enrich it. It's in the form of O18 water, right? So we have about a couple mils of water, and we're shooting protons at it. Now, the nucleus itself is small relative to the atom, so it's mostly space, right? So more often than not, even though you're shooting a proton at it, it's going to miss, right? And it might even just miss by a little bit. And when it does, it's going to just deflect off. They're both positively charged, so it goes off to the side, and ultimately it's usually dissipated in heat. If you're shooting it and you happen to be directly on target, but it's not energetic enough, it's going to get closer and closer to the target, to the nucleus, but it's just going to bounce back because the force of repulsion gets higher and higher and higher. But if you get it to high enough energies, which is like on the order of 10 to 20% the speed of light, you can actually get it to go into the nucleus itself. And if this is O18 and we're making F18, what ends up happening is this goes into the nucleus, it ends up expelling a neutron, and you end up with F18. This is how you make oxygen 15. Oxygen 15 has a half-life of about 2 minutes, and we make this a couple times a week at the University of Iowa to do research brain blood flow studies. We don't accelerate protons, we accelerate deuterons. We use N14 as a target. When it goes into the nucleus and it gives off a neutron, we end up with oxygen 15, which is a half-life of just 2 minutes. There's not a lot of chemistry you can do on something that has a half-life with 2 minutes, but we can make water with it. So we make O15 water, which we use for quantitative brain blood flow studies. How many mils per minute are actually going through the various different parts of the brain by doing. So anyway, this is just cyclotron production. Here's, I think, 2, 4, 5 of the more commonly used radionuclides out there. So fluorine-18 we talked about, half-life of 110 minutes. O18 is the target on that. Carbon-11 has the half-life of 20 minutes. Oxygen-15, 2 minutes. Zirconium-89, half-life of 78 hours. Why would we inject something radioactive into somebody that had a half-life of 78 hours? Well, sometimes we want to do things like probe the immune system with monoclonal antibodies. Monoclonal antibodies have to go around the system for days before it localizes where it needs to. So you can't put something short-lived on it, or you're not going to be able to see anything by the time it gets there. So we have to tune the half-life of the radionuclide for the purpose that we're actually imaging. I think I'm going to skip this. We have to be careful about what radionuclides we choose. It's not just the half-life that we worry about. It's can we actually produce it? It's are there additional gammas? Sometimes that's a good thing, sometimes it's a bad thing. What are its chemical properties? If I made a really cool radioactive neon, it's a noble gas. It doesn't react with anything. I can't add that to anything, right? So you have to put all these things into the recipe for these things. So sometimes the half-lives are too short, sometimes they're too long. There aren't that many left, but there are enough, and we can do amazing things with that. Okay. Real quick, I want to talk about the diversity of PET radiopharmaceuticals that are out there. As I said, thousands have been tested. There's about 15 or so now that I think are approved for use clinically and are reimbursed. But the first thing I need to point out is, I mentioned the FDA. So because we are manufacturing drugs, much of the synthesis has to be done under aseptic conditions. So that's number one. Number two, we're dealing with radioactivity. And we're generally, when we're making this, we're making it for a number of patients, and it's decaying. So we have to try and make as much as we can so we can inject as many patients as we can. So everything is not only done under aseptic conditions, but they're done remotely by remote synthesis units and by computers in hot cells that have 3 inches of lead in them because we have so much radioactivity in them. So this is it's a regulatory nightmare, but we do it anyway, and it's hard and it's expensive, and the regulations keep on getting more strict, but we're persevering. This is just, there's 2 slides here. I'm just going to briefly go through just to give you an idea of the breadth of what it is we do with PET clinically and research-wise. So we talked about the glucose, methionine we use for brain tumors, N13 for cardiac blood flow, oxygen-15 water we do for brain blood flows. I'll show you some images of gallium-68 dotatate, which we use for neuroendocrine tumors. There's these several PSMA agents, gallium-68 PSMA and others for prostate cancer imaging, fluorethymidine, that's for cellular proliferation, fluoridopa, Parkinson's disease, zirconium-89, that's just one of the many antibodies that we've labeled, and antibody fragments. Zirconium-89, the kinetics are long, hence zirconium-89. Other good ones here. Florbetapir and flutamidamol. These are amyloid imaging agents for Alzheimer's disease, so we can diagnose Alzheimer's disease with great specificity and sensitivity. We'll talk a little bit about, well, we won't talk about, but raclopride, for example, that's just one of the receptor systems in the brain that we can study. So dopamine D2 and D3 receptors. Sky's the limit on what it is we can do here. So now I want to show you some examples of some of these. So this is a prostate cancer imaging agent. PSMA, prostate-specific membrane antigen, is expressed on the surface, on the cell membrane of 95% of prostate cancers. And so we have developed molecules like this one, and it's this part right here that actually ends up binding to this PSMA with great specificity. So all these dots here are metastatic prostate cancer tumors that are kind of all over the body. I want you to note a couple of things. One is, unlike FDG, this is really specific. It goes almost exclusively to the tumors. A little bit in the liver there. The kidneys I don't worry about, and the bladder I don't worry about, because that goes in and then it comes out. So it doesn't stay there for very long. I know it looks like bad. You're a little bit more worried about the salivary glands, which makes it not very prostate specific, but nonetheless, it's very specific for the cancer itself, and that's going to be important later. So that's not the only prostate cancer imaging agent that's out there. They've looked at all of these that you can see here, right? And all of them, however, have that same chemical unit hanging off that are all circled there, and those bind. This doesn't get small enough. You'll actually see there's a little red dot right there. This is the prostate-specific membrane antigen. That's the cell membrane there. These bind very specifically and avidly and long-term to that little spot right there. That's what makes this all work. So understanding the biochemistry of the body, engineering the radiopharmaceuticals to go there. All these other things that are hanging off there, because there are subtle differences in how well they bind or how easy they are to synthesize, right? There's all sorts of different kind of subtleties, so we keep on trying to find the very best ones. Okay, here's gallium-68 DOTA-TOC. That's what we use at the University of Iowa to image neuroendocrine tumors, and copper-64 DOTA-TATE, and that's on the left. It's this area down here that binds to the somatostatin receptors that are overexpressed on the cell surfaces of neuroendocrine tumors, right? Now, I show these two because we use gallium-68 because it's relatively inexpensive for us. It has a half-life of an hour. It's no big deal because our chemistry manufacturing area is just across the hall from the scanner. But if somebody in Idaho Falls has a neuroendocrine tumor and they need a scan, and the nearest cyclotron is 400 miles away, how do you get it there, right? So copper-64 has a half-life of about 12 hours. And so there is a manufacturing company in St. Louis that makes a bunch of this stuff every night, and they send it overnight. It's 12 hours is long enough that you can kind of overnight it, even sending on commercial airliners, and get it to Idaho Falls and get it into the patient. So functionally the same molecule, but different radionuclides because we have different delivery purposes for it. So here's what a copper-64 DOTA-TATE scan looks like, and a gallium-68 DOTA-TOC. Once again, highly specific for the tumors, and that's going to be important later. We also can help with breast cancer imaging with fluoroestradiol. It's an estrogen receptor. Some breast cancers express estrogen on the surface, And those are actually treatable and treatable benignly with hormone therapy. And so if your breast cancer expresses estrogen and only, and those cancer cells, all of them, the tumors, only express estrogen, they are candidates for this particular hormone therapy. If some of them, if it's differentiated to the point where some of them do and some of them don't, you don't want to treat them with only hormonal therapy because the other ones are going to end up untreated, right? So you need to somehow be able to help the physicians guide how they treat these patients, and fluorestradiol does that in breast cancer. Amyloid imaging, you can see here, there's 3 approved agents for that. I'll just simply say these are about 15 years old, and they weren't used for most of that time because we got them approved back 2008, 2010, 2011, and Medicare decided they weren't going to pay for them. So FDA approved it, and they said no because there were no treatments for Alzheimer's disease. But now there are. And so over the last year or so, we've started doing a lot of Alzheimer's disease scans with these drugs that have been hanging around for a while. Everything I've talked about so far are approved drugs. This is probably one of the next on the list. These are fibroblast activation protein inhibitors, and there's a bunch of different ones that are out there testing. We're not imaging and binding to the cancers themselves. We're binding to cells that form around cancer cells. So to the fibroblast, so this is the tumor microenvironment. It's targeting the tumor microenvironment, not specific to a particular cancer, which is a good thing. Sometimes being specific is good. Other times, but being able to diagnose colorectal cancer, pancreatic cancer, non-small cell lung cancer, breast cancer, ovarian cancer, all with equal specificity, that's a good thing. And it doesn't have all that background activity that glucose does. Glucose is general too, but it's a lot harder to interpret. So these are in late stage clinical trials now. I expect in 3 to 5 years we'll see this in common use. So keep watching this space. And then I've ignored a bunch of stuff. I'm not going to go through this. But PET is playing a big role in a lot of drug development space and immunotherapy drugs. There's a lot of neurological applications and drug development that I didn't go into. And I didn't even touch on cardiac work, and there's a lot of cardiac PET that's going on too. Okay, going to move on to PET scanner design, how all that works. So before we understand how a PET scanner works real quick, we need to understand gamma interactions with matter. Once again, it's not that hard. Because there's easy stuff, like it might not interact at all. It'll go right through the body. And in fact, that's best case, right? Because we want to be able to detect it. But the other thing that can happen is it can go in and it can have a photoelectric interaction. That's when it interacts with an inner shell electron and the entire photon is absorbed and we just don't get anything out, right? That doesn't happen very often in the human body because it's proportional to Z cubed, which is number of protons in the nucleus. We have low Z material in our body, carbon, hydrogen, oxygen, that's low. But what does happen a lot is Compton scattering. And that's when a photon goes in, it hits an outer shell electron, and then there's an inelastic scattering event, and so the electron kind of goes off, doesn't travel very far, and the photon goes off at a different direction, which is a problem because we depend on that collinearity, right? And the energy changes. So it goes from 511 keV to, let's say it's a 350 keV, because depending upon the angle it's scattered at. So 2 things happen. And this happens a lot. In fact, the probability of both 511 keV photons escaping the body unscathed happens only about 5% of the time, right? I wish it didn't, but we really only get 5% of those because of the Compton scattering. So we have to take all these things into account. So how does this whole thing work? We saw this before. I'm going to go into a little bit more detail here. Tens of thousands of little crystals. We'll see those in a second. The scanner's designed to simultaneously detect those two, once again, within about 5 billionths of a second. When an event is recorded, we record the individual crystals that the event occurred in, and we record the time at which it occurred to within less than a billionth of a second. So when these happen, there's a lot of right into the hard disk that's going on here in a really rapid fashion. So the pipeline for this is really fast. And in about 10 to 20 minutes, we get these tens of millions of photons, and then we reconstruct. And we talked about that before. So there's a couple things that can happen here, but first let's talk about the PET detector modules. How do these PET detectors work? I talked about scintillation crystals, right? So tens of thousands of them. Each one is usually about 3 millimeters by 3 millimeters by 20 millimeters long. That's the primary limit to our resolution, is the size of our detectors, and we don't want to make them much smaller than that. A scintillation crystal is a special material that when a high energy photon hits it, it creates a flash of visible light. So one high energy photon, thousands of visible light photons, really fast. So you get this flash. That little flash of light, it totally internally reflects inside that crystal. It hits the photomultiplier tube. A photomultiplier's tube's job is to turn visible light into an electrical signal. And the signal that comes out is proportional to the amount of light it detected. The amount of light you had is dependent upon how much energy went into that gamma ray in the first place. So that pulse you get out is indicative of the energy of the photon that interacted in the first place. Okay? So let's go back to this. So what ends up happening is a photon from crystal A absorbs the gamma ray. You get a flash of light. That flash of light turns into a signal. That signal goes to an energy discriminator, which you see here. Is it 511 keV? I'm good with it. Then it sends out a fast pulse. At the same time, same thing's happening with B. You get this, you get the flash of light. You get the absorbed photon, you get the flash of light, you get the signal. Is it 511 keV fast signal? And if these two fast signals hit that coincidence box within 5 nanoseconds, within 5 billionths of a second, it's an event, and then we record it. Okay? We record which detectors at what time. If two events happen at different times, they may both be 511s, but they won't hit that coincidence box at the same time within that 5 nanoseconds. It'll hit one, but not the other, so you just throw it away. Another thing that can happen is scattered photons. Okay? This is where one makes it out fine, but the other one scatters. So the one that makes it out fine gets this nice big 511 keV photon, and we send that fast pulse. The other one is, let's say it's only 250 keV. It hits that crystal, you get a flash of light, but it's a smaller flash of light, smaller energy, smaller signal, and so this energy discriminator says, not happening. And so you don't send anything out, so we don't count anything. So we can discriminate on that scatter based on the energy of that detected photon most of the time. One of the more interesting things that can happen is an accidental coincidence event, where you have two individual events, but they happened at the same time. So you detected one, you detected another, the other ones you didn't detect. They both send these pulses out. They pass the 511 test. They both go into the coincidence box at the same time. So you say, I'm going to count it, but we're recording that crystal, and it says, ooh, the event happened along here, and it didn't. So we got a problem, right? So we handle this, and we can actually handle it quite well statistically by measuring the count rates of all of the detectors. We can, to within a couple percent between any detector pair, predict about how many of those we're going to get based upon the count rate, and we can subtract those off, and we can handle it. But we have to handle it because this happens a lot. And the more activity, the better, or the bigger the problem is. Okay. For about 32 or 33 of my years in the biz, we use these photomultiplier tubes. None of the scanners being developed today use these anymore. We've changed technologies. The photomultiplier tubes are out the door, and the silicon photomultiplier arrays, which you see down here at the bottom, have replaced it. They do even a better job of converting light to electrical signals. They do it faster. These are smaller elements, so we can determine which crystal the event occurred in with much greater accuracy. We can actually use smaller crystals with these things, and we just simply get better images. And they're fast. And that fast is going to come into play real quick. Each of these squares down here at the bottom, which is the light detector, it's going to convert it, actually consists of an array of thousands of these little microcells that are about 10 micron. That's like twice the size of a red blood cell. Each of these are light-sensitive avalanche photodiodes, such that when a light photon hits it, it sends a little current on through. But there's tens of thousands of these. So when the light from the scintillator hits it, you get the flash of light. The flash of light turns that into the current. That's the current, which ends up being sends the signal, which is proportional to the energy, and it goes on through. So it does everything that the photomultiplier does, but it does it better, and it does it faster, and it does it more accurately. I just want to bring your attention. I've been mentioning PET scanners, PET scanners, PET scanners. You can't buy a PET scanner anymore. You can only buy PET CT scanners. This is an article from Time magazine in the year 2000, where the PET CT scanner won Invention of the Year. I didn't think it should have won Invention of the Year. I mean, it was groundbreaking out, but it was really just bolting a PET scanner to a CT scanner. I guess it's like chocolate and peanut butter. It's like a Reese's peanut butter cup. I mean, somebody's got to do it. But it did revolutionize it because it actually allowed radiologists to read this. Usually nuclear medicine physicians would read these PET scans. This enabled radiologists to actually read it better because they actually had this anatomical backdrop, which helped a lot. So it is a big deal. I don't mean to downplay it. Okay, image reconstruction, time of flight. I just want to give you a feel for the fact that we actually can reconstruct these images, not knowing exactly where it happened across the scanner. So what we have here is a ring of detectors, which you see here, with just a very simple, almost point source in there. And so what I want to do is look at the detector pairs that happen to be just horizontal across there. And most of them are going to detect 0, 0, 0, 0, 0, 0, 0, until you get down to where the middle, where the activity is, and then all of a sudden you detect a lot, and then 0, 0, 0, 0. And so you can do a projection along that horizontal line of where the activity was. That's that horizontal line that you see there. If we look at 45 degrees, that's what that 45-degree thing is there, and where they overlap, it's kind of turning yellow there. And if you look at 90 degrees and 45 degrees, you put those all together. I mean, it certainly doesn't look like that beautiful dot in the middle, but you can kind of see how we're kind of sort of getting there, right? And that's just 4, right? What if we did 16 projections? Or what if we did 128? It kind of looks like it, but you have that kind of haze behind it, right? It turns out that haze has a mathematical shape to it. It's really just a 1/r curve. We can take care of that mathematically. We can remove that 1/r blurring. And we do it in Fourier space. The math is really fast there, and so we can just literally get back to what we started with. That's a simple case. This is a more complicated case in the upper left-hand corner. That's actually a PET scan. And we're just going to look at this from all sorts of different angles. So it's projected this way, that way, this way, that way, this way, that way. And if you look at all those different projection angles and you add them all up, superimpose them just like we did in the other one, you get something close. It's just that really blurry image. It has that haze just like our other one did. But if we go into a Fourier space and we filter it, and then we reproject it, you can actually get it back exactly. This is the same thing that CT scans do. We're doing the exact same math, the same everything. Just taking this projection data and making tomographic images from it. So now, just real quick, this is, well, I should say it was kind of a revolution starting probably about 10 or 11 years ago, where we started doing time of flight PET. Now, actually, time of flight PET was actually a big thing back in like 1991, 92, but we kind of sucked at it. Our crystals were fast, but they weren't dense enough. Our electronics were too slow. The computers were too slow, and it was one of these things that the time just hadn't come. But then technology got better. We got better crystals. We got faster electronics, everything. And all of a sudden, now what is time of flight? Time of flight is, it used to be, and the way we've been talking about this, is where did that event happen? It happened with equal probability somewhere along the line there, right? But what if we knew that it happened in that detector down here? It got here first and got there second because it was closer. Now, the speed of light is pretty fast, but our electronics is getting pretty good. So if we know that it happened there first, now we kind of have a probability distribution. And that width of that probability distribution has to do with how good our timing resolution is. And so if we're at like 600 picoseconds, we can get it down from across here to down on the order of 9 centimeters or so. If we can get it down to 500 picoseconds, we're down to about 3 inches. 300 picoseconds was a pipe dream a few years back. That's down to like 2 inches. And we're installing a scanner right now at the University of Iowa. We're testing it as we speak, hopefully going to accept it pretty soon. We're down to 178 picoseconds. We can localize it to within an inch before we do any of this reconstruction stuff. So our reconstructions get way, way better. So this is just with the standard equal probability, a little bit more complicated. You can see down there. But if we have time information, and this is just with 4 projections, you can reproduce it easily. And it makes our images just simply better. The signal noise is 5 to 10 times better with 178 picosecond resolution than without any time of flight at all. So this is the way virtually all the scanners are moving right now. I need to spend just a minute, because this is what I spent most of the last 20 years doing. These PET scans aren't just pretty pictures and diagnostic pictures and all. They're quantitative imaging devices. They're exquisite quantitative devices. Each voxel, each pixel in this image, I can tell you how many decays per second are occurring in that to within about 2 to 3% if I got my scanner calibrated accurately. I mean, this is crazy good, right? We can do all sorts of pharmacokinetic modeling with these things, and we do, but it's kind of gone clinical. But this has kind of been lost in all of it. These were always measurement devices, and they continue to be, and they're crazy good. Okay, some quick new developments in the field. This is just kind of showing what brain scans look like. These are all FDG brain scans starting in 1980, going to 2024. Now, this is a new scanner. It's one of a kind right now, a colleague at Yale has, where we're down to spatial resolution of this brain-only scanner of on the order of about 1.5 millimeters. That's like MRI type reconstructed resolution. Never in my life did I ever think we'd get here, because I started over here. So it's crazy good. This is a picture of that scanner. So Rich Carson at Yale worked with United Healthcare Imaging to make this first of its kind system. I won't get into it, but they have some technologies that allow for smaller crystals to be used. I didn't get into it, but one of the challenges when you get smaller crystals, that's the reason you can't get too small, is because these photons can actually go into a crystal not straight up and down, but obliquely, right? So if it hits this crystal, it might go through until the next crystal before it actually interacts. You get the photon, and it'll misplace where that photon interaction was by like 3 or 4 millimeters, and that'll decrease the resolution. They're using some depth of interaction technology here, so you can tell not only which crystal, but how deep in the crystal it was, so you can much more accurately define that line of response. And it buys you crazy good imaging. I don't know whether you can see it from here, but those on those little pie-shaped things, the smallest one, functionally 1 millimeter little dots of radioactivity separated by 1 millimeter, and you can resolve those. I mean, that's amazing. And that's an FDG scan. This is an FDG scan compared to an MRI scan. Now, MRI, if you really want to, you can get sub-millimeter, right? But this is amazing stuff, and it's going to enable some amazing neuroscience. Is the deal? This is not a clinical machine. This is a research machine. Going the other way. These are a couple of friends and colleagues of mine, Ramsey Badawi and Simon Cherry. They work with the same company, United Healthcare Imaging. They're at UC Davis. They built the world's first total body PET scanner, 2 meters long, 500,000 of these little detectors as opposed to on the order of 20,000 for these. Normal PET scanner, you can image this much at a time. Here you can image the whole body at a time. You get a whole image in a minute or two that would knock your socks off, like this. You could use this for a high throughput scanner and just run patients through as much as you want, but that would be kind of a waste of, well, maybe it's not a waste of the technology. But you can do some amazing things like this with whole body pharmacokinetics. So this is one of the early images that came up, and this is going to keep running, so we're going to talk about this just for a second. This is a glucose scan. This is an amazing thing to be able to see, second by second. So they're in the scanner, you inject the FDG, and they're taking a whole body scan every second. And so you can actually see this movie. So we're going to wait till it kind of starts up. You'll watch it go through Normally we inject in the arm vein. They inject it down here, so you can actually see it go up. You're going to see it go into the heart. Now it's going to go into the lungs. Now it goes into the major arteries. Once it gets to the capillaries, it can actually go into the tissue. See it diffusing in the tissue. Now, if you watch the brain is metabolizing glucose. We're only 2 minutes in. Usually we wait an hour before we do an image, but you can see it concentrating more and more and more in there. This next time around, I want you to watch the kidneys. So it's going to go in. The kidney is the only organ in the body that can recognize glucose as an imposter, so it filters it out. So you can see it go into the kidneys and then go into the bladder in real time. And there it goes. And that actually helps clear out some of the background activity there. Amazing things to do. This is very simple kinetics, but we can do whole body kinetics on this whole range of radiopharmaceuticals that we have. Okay, last bit here. I'm going to try and go fairly quickly, but this is where the field of nuclear medicine is going with a vengeance, right? So this is a PSMA image on the left. It's a dotatate neuroendocrine tumor image on the right. I think you've seen the one on the right before. And I talked about how specifically these PET agents go to these tumors. So we go to great lengths to inject as little radioactivity as we possibly can in these patients so they don't get needless radiation exposure and still get killer images and they're diagnostic and everything. But it makes you think. We know that very large doses of radiation can kill cells, or at least damage the DNA so they can't reproduce anymore. And that's what external beam radiation therapy is all about, right? When people have cancer, it's localized in the lung. That's what's happening in this patient. You blast X-rays through it, a particular sublethal amount of radiation through them, right? But some of it goes through the tumor. Then you do it from a different angle, sublethal, but it overlaps, right? And then you do it at a third angle, sublethal, and it overlaps some more. You get a lethal amount. You can measure the amount of radiation dose they get, and you can actually kill the tumor while you get a fair amount of radiation to the tissue surrounding it. It's not lethal and not toxic, and you can treat cancer that way. So it makes you go, what if we didn't just inject a little radioactivity into the patient with cancer with one of these radiopharmaceuticals? What if we injected a lot? Well, you wouldn't want to do it with gallium because it has to have half-life of an hour. So let's choose something with a longer half-life, like lutetium-177. It's a beta emitter, has a half-life of about 8 days. These things bind really tightly to these binding sites. They'll last there for weeks, right? So you inject lots of radioactivity. And so this is the PET scan, gallium-68. You can see tumors everywhere, right? So now they've injected the patient with a lot of lutetium-177 dotatate. Now I showed you that energy diagram before. You get some gamma rays from that, right? So we can image it. Not with a PET scanner, because we don't have any positrons here, but with gamma cameras. That's another nuclear detection device, so we can image it. They're not as high resolution, not as good with the quantitation here, but there it is, and you can see it, and it's the same distribution, just like you'd expect, because it's functionally the same molecule. And it sits there irradiating the tumors for days. And it can kill the tumors. Now, this is a super responder. Not all patients end up like this, but this is kind of proof of principle, and this stuff really happens. So this company that got this approved in 2018, AAA, Advanced Accelerator Application, small French company, got this approved and were promptly bought by Novartis for $4 billion. And then all hell broke loose. Now, instead of 2 or 3 companies doing this, there are 150 companies doing this. This is all the clinical trials that are going on right now. It's the reason I was here, because we're trying to figure out how to do these clinical trials faster. Because there aren't enough sites to do these studies who know what's going on. So, but it's a crazy, crazy good thing. So that's just the gallium and the lutetium agents. Now, neuroendocrine tumors are a rare tumor. Prostate cancer is not. This is the one that you see on NFL football games. The same company, Novartis, bought the IP for this agent, which is prostate cancer. This is 6 cases. The red ones are the first before therapy, and then So this is before therapy, after therapy, before therapy, after therapy. And once again, this doesn't happen on all patients. But this is kind of a remarkable thing. Very, very specifically treats the prostate cancer. But sometimes it doesn't work, right? But I know why it doesn't work. You know why. This is not some funky, bizarre immunotherapy thing that nobody really understands and can predict. The tumors didn't get enough radiation dose, right? Let's just inject more. Well, you can't just inject more. And you have to worry about radiation toxicity associated with it. But you know what? We can image it, and we can quantitatively image it. We actually might be able to measure that radiation dose. We actually might be able to titrate the amount of radiation we give these patients specific to that patient. Right now, we just give 4 cycles of 200 millicuries blindly every time. And sometimes it works great, and sometimes it doesn't work at all. What we like to do is to use SPECT imaging to measure the amount of radioactivity in the organs, because we have to be careful about toxicity of the organs, right? We'd also like to measure the dose to the tumors, just because we need enough to kill the tumors. So we'd like to quantitate that. We could use this and we could titrate. Now, the little asterisk down there, we're just not very good at it yet. This is a hard problem, but we're getting there, right? So this is what we're working on now. So the way this works is you inject a large amount of radioactivity into a patient, that's lutetium-177. You can image them at 4 hours after injection, the next day, 48 hours, 72 hours. You have the activity, how many decays you get in the organ, in this case, let's say the kidney, right? Over time, you know the energy per decay, you know the time. You can actually calculate how much energy is being deposited in the organ or the tumors, right? It's not hard as long as the quantitation is good. So what one would like to do is not just do that standard 200 millicuries. So the FDA has limited us to 23 gray to the kidney. That's a unit of radiation dose, 23 gray. We need to stay below that or else you might have kidney toxicity. It's probably closer to 30. They got it wrong, but I'll even live with the 23 for now. So what you would do here is you'd give the patient their standard 200 millicurie first, you'd image them, you'd measure the radiation dose, and let's say the kidney's got 3 gray. Well, if you did that 4 times, you'd get 12, which is about half of that 23. We could really bump this up. So the next time, you give them 400. And you don't just guess, you actually measure it, but let's say you get the expected 6 gray to the kidney, and then you do it again, 6 gray to the kidney, so now we're up to 12, 15, you do it again, you're up to 21, you're still below. You've given the patient almost twice as much therapeutic radiation. The probability that they will be a responder and respond more is a lot higher here. But we have to be able to do this and do it accurately and reproducibly. We're not there yet. And we're probably 5 years away, I'm guessing, until we're there. But we're all working together on it. Everybody thinks this is important. Industry thinks it's important. Pharma thinks it's important. The scanner manufacturers, academia, the doctors, the FDA. The FDA is working with us on how to do this, right? So I'm confident we're going to get there. This is where it's really getting exciting. And the other place it's getting exciting, this is the last slide, and I'm sorry I've droned on this long, but here's a case where we have a patient that was given lutetium-177 PSMA a couple times, and it wasn't working. You see, it's just getting worse. So they injected the patient with the same PSMA, but labeled with actinium-225, an alpha emitter. And you get sometimes remarkable responses like this. And this is not uncommon, but we don't understand a lot about alpha toxicities and things like that. So it's getting kind of tricky, and we got to watch it. So thank you all for your attention. This has been fun.
So we do have time for questions.
My name is Al Ehrlich. I am a member of the PSW. The 2 questions, if you don't mind. One is these detectors. I did some of this stuff 50 years ago. Back then, sodium iodide, lithium-drifted sodium iodide, stopped the gammas quickly, thoroughly, but not quickly. So you couldn't use them for fast timing. Are those crystals now totally outdated? Those kind of crystals?
So sodium iodide crystals are outdated in the PET biz. So, and the PET, when I was in graduate school, they were all sodium iodide, and they were slow, but we weren't doing time of flight. But until probably 1990-ish, they were still sodium iodide, maybe a little bit earlier. Then they went to bismuth germanate, BGO. But standard gamma cameras still almost exclusively use sodium iodide. In PET scanners these days, and everything was too long. I had a list of the scintillators that we were using in PET. These are lutetium orthosilicate, are the crystals we're using now. They're very fast, they're very dense, and they have pretty good energy resolution. So they check most of the boxes. They're not the best, but they're
What
good.
are they made of?
It's lutetium orthosilicate, sometimes yttrium doped, sometimes not.
Okay. Second question, totally different. What is it is there any understanding of what it distinguishes the good responders from the non-responders to the therapy?
So a lot of it has to do just simply with the density of the receptors on the tumors, right? So if they're sparsely on there, then there's not many receptors, and so you don't get as many radioactive atoms on it. That's one thing. There are other things that impact radiation sensitivity, like how much oxygen the tissue gets. So there's a lot that has to do with the tumor microenvironment as well. But mostly it has to do with how avidly the particular tumors will bind the tissue, and that has a lot to do with the receptor density on them.
Thank you very much.
Blue microphone.
Hi, Matt Kaywood, member. In neuroimaging the brain, having structural images can often help you reconstruct, solve the inverse problem and identify sources, right, very accurately. I don't think you talked too much about this, but how, in current practice, how important are the CAT scans or other structural scans or MRIs for that matter in getting the resolution that you want, and are there gains in the future that can be gotten from having better structural imaging?
Yeah. So the gains that you get from combining the information from, in particular, MRI scans for resolution purposes are in the research domain. So you kind of force the PET images into the higher resolution MRI, and you try and at the same time maintain the quantitation, and that's the challenge. But that's mostly in the research domain. That's not so much used with the CT scans. The CT scans are more often used for anatomical correlate. And one good example of that, for example, I think I showed a prostate cancer patient that had a CT scan. They had one little dot there. So it turns out you have ureters that kind of go down and the kidneys filtered out. Sometimes there's little dots of urine in the ureter, which can just look just like a lymph node. But if you have the CT scan, you can say, oh, yep, that's a lymph node or that's the ureter, and it'll help you differentiate and not misread.
So, but you said it was in the research domain, so presumably researchers think there are some gains.
Yes,
to be had there?
yes, absolutely, but largely in the neurospace. But yes, absolutely.
We'll go to the web. If you have a question, please keep your hand in the air long enough for the mic runner to get a mic to you. You have to keep doing that over and over again until you get a microphone. So do we have a question from the web back there?
We do have two questions from the web. The first question, well, Joel actually has two questions. So I'll ask Joel's first question. In a previous talk on LLMs, it mentioned a problem of knowing what constitutes good data. If you made a vector space where one axis is MRI results, another X-rays, another PET, another CAT scans, could you use AI to get a better analysis of the problem or medical issue?
So the answer, I think, is theoretically, the answer is absolutely yes. Trying to get enough data to, I'll tell you what the problem is in the whole medical imaging space as far as AI is concerned, is it's all about HIPAA. The ability to share medical information with patient information. The universities and the hospitals are really run scared on that. So you need large data sets in order to do this, and they need to be well curated. and nobody's willing to give their data to create these large data sets where you can actually make these correlations. So theoretically, absolutely. In practice, people are actually doing things like this, but they're doing it in their own institutions with their own equipment, and it's not really generalizable, at least in the near term.
The second question Joel has is, has there been any use of PET scans in veterinary science treatment of animals?
And the answer is absolutely. There are PET scanners at veterinary schools, and largely in cats and dogs for cancer diagnosis. And so it is done. It is not common, but it is becoming increasingly common. And what often happens is a hospital PET scanner will kind of get old and out of use, and the veterinary schools can actually buy them and operate them. This is the way it works.
Okay, and the last question is from Eric, who's streaming from Sweden. Eric asks, has anyone combined a PET scan with the MRI yet?
So yes, and in fact, when I mentioned that PET scanner that's being installed at the University of Iowa, it's a PET MRI scanner. And they've been around for on the order of 15 years or so. This is kind of the latest and greatest, and it has really good timing resolution. There are some reimbursement challenges with them, and as such, and I won't get into that, but they do have applications. They have neuro applications in Alzheimer's disease for better diagnosis with combined PET and MRI, and also prostate cancer is another good one. We're going to be using it for multiple myeloma as well, because you can get complementary diagnostic information of the two, and the correlation, the exact correlation of the pathology is important.
Thank you.
I believe there's a blue microphone question somewhere. Yes, can you stand up? Please tell us your name, and I know you're a new member, so welcome.
Joseph Crandall, new member. You were discussing the progression of time of flight resolution in the picoseconds. Do you know any limits to pushing that to the femtoscale? And if not, what new applications will that unlock?
Okay, so yes, there are limits, and I shouldn't be saying. I mean, I didn't think we get. If you would ask me, I wouldn't have expected we could have gotten to 178 picoseconds in a routine machine. But what you do end up with is you have not only do you have the timing resolution of this, but the photon then goes into the crystal, and you have the distance it travels in the crystal before the photoelectric. So you have other timing things that are now on the scale of what it is you're trying to measure. So a 20 millimeter long crystal where that photon interacts and when it interacts is of the scale that we're talking about in the body as well. So you could maybe start making your crystals smaller, but then you lose sensitivity, and so there are trade-offs there. But there are people who are thinking outside the box on how to do this even better. But I'm just thrilled at where we are right now. I'm happy as a clam.
I have a blue microphone question back there in the back of the room.
Yep. Adam Jacobson, PSW member. A few times you mentioned the 5 nanosecond limit for counting two events as being the same event. Could you say a bit about where that 5 nanosecond number comes from, and if that's changed or will change over time as detector technology improves?
So it won't change that much. It might change a little bit. So here's the deal. So we need to be able to handle the situation where an event happens on the very edge, where you have a short distance and a long distance. And so that's about, and it's about a 70 centimeter axial field of view. So that's about 2.5 nanoseconds. So you can't do any better than that, right? Because you have to be able to cover the whole area there. The other couple of nanoseconds has to do with signal transmission through the wires and stuff. And there are different lengths and things like that, and they do their very, very best to compensate for those. But that's where the other half comes from.
Thank you very much.
Red microphone.
Hi, John Chernyovsky. I'm a member. I have prostate cancer, so I have a great deal of interest in this new technology. One of the things that I've heard about this particular radial pharmacology sort of application is that there are other parts of the body where the PSMA scan will say it's active, and you'll get the radiation where you don't really want it. In particular, I believe the salivary glands and
Yep.
other parts.
Yep. So what we're talking about here is not so much in the imaging. It goes there in imaging and it goes there in therapy as well, right? But you saw in those images, you could see it was going to the salivary glands and the parotid. And if you get too much radiation there, then it can functionally kill the salivary glands. And so when you eat from there on out, you're going to have to kind of be sipping water as you're doing it. And it is one of the side effects that they watch out for. Not everybody has it. It's one of the things we could probably predict with dosimetry, but we don't. So with most organs, we know what the radiation limits are from external beam radiation therapy, at least ballpark. The salivary glands and parotids, they're reasonably well studied, but not particularly well studied. So that is one of the few significant side effects that these radiopharmaceutical therapies have, and that's specific to the prostate cancer treatment.
Okay, thank you.
But you're dead on. Yep.
Blue microphone, Tim.
Thank you, Timothy Thomas. I am a member. I'm asking about the possibility of using PET scan as improvement for functional MRI. You've talked about medical procedures, but what about using it to more accurately pinpoint activities of the brain, for example?
So we were doing that with PET back in mid-'80s. We were doing it with FDG, fluorodeoxyglucose, and we were doing it with that O15 water. A lot of the brain mapping, so MRI has taken over a lot of that since then, because MRI can do that, but PET did it first, and we did it well. So you would actually have people performing functions in the scanning. That's where we actually learned where the visual cortexes were, where the auditory cortexes were. There's some great papers by John Mazziota, where it kind of shows, here's speaking, here's doing math problems, here's frontal cortex. So yes, we've done that, and we continue to do those things. In fact, we're doing some research studies right now on patients with cochlear implants to try and figure out when you start hearing better with cochlear implants, what ends up being activated now that wasn't activated before, so before and after cochlear implants. They can't do that with MRI because of the metal coils, right? So it's kind of niche. That is one of the things that I actually want to do with the PET MRI is compare the functional, the brain mapping in the same patient at the same time, comparing O15 water blood flow with what they're doing in MRI, and I think there are going to be some differences, frankly. And ours is right.
We're going to take one from the web, and then we'll go to Jared with the blue microphone.
We have another question from the web. They did not give their name, but the question is, are you integrating PET with brachytherapy, I guess targeted radiation therapy?
Well, not with brachytherapy per se. So brachytherapy, for those who are unaware, is, let's say you have prostate cancer, they will actually physically embed a radiation source in the prostate to expose it directly. So that's brachytherapy. We are not, to my knowledge, I don't know of anything that's doing that, but we are combining this radiopharmaceutical therapy with external beam therapy. So actually that's kind of all the rage right now. We're taking these approved therapies and we're combining them with other therapies to make them more effective. So radiopharmaceutical therapy with immunotherapy, radiopharmaceutical therapy with external beam radiation therapy, et cetera. And these combined, this is where we're going to see the near-term improvements in therapy efficacy.
I would like to go to the red mic if it's okay. Red mic?
Oh, I'm over here. I am not a member. My name is Chanel. I would like to know if injectable insulin would serve any purpose in this process.
Can you hold the mic?
Oh, sorry. I'd like to know if injectable insulin would serve any purpose in the process of the glucose uptake.
Okay, so that's actually a very interesting question. So when we image patients, we want their blood glucose levels to be low. So we have them fasting for 4 to 6 or 8 hours before scans. So the blood glucose levels are low so that it's not, the glucose we inject is not diluted, right? Because if you have a bunch of regular glucose there that's otherwise diluting it, the uptake won't be quite so high. And so what we end up doing is trying to control the glucose. It gets tricky with diabetics, and if when we do have diabetics, then we do insulin protocols in order to kind of make sure their blood sugar is down. And so we do that not routinely. We try to do it dietarily by keeping the blood glucose levels low. That's a really good question.
Blue microphone, and then we'll have a comment coming in from the web. Blue.
Great lecture. My name is Jared McQueen. I am a PSW member. You mentioned the HIPAA hurdles. There are some other challenges like the Vesuvian scrolls, those ancient scrolls, where they open sourced a gigantic data set, and a lot of scientists from around the world were able to decode the contents of them. If you could have like an organ donor equivalent of these patients where they're like, I want all of my data online, and would it potentially unlock maybe new methods or new algorithms for detecting these things?
So that's a really interesting question, and it's multifaceted, right? So one of the things we've talked about doing at the university, it doesn't get a whole lot of traction, but when we have patients come in, one of the things that we've talked about is having patients sign kind of a universal consent that we can at least use some aspect of your medical data completely de-identified for research purposes unencumbered. And that would be great, and I mean, I would sign something like that, but not a lot of people would. But hospitals are really, really, really risk averse, and I don't know of any institutions that have gotten any traction with that because it's a relatively common idea that makes a whole lot of sense. But
Well,
sometimes,
forget it.
Yeah. Well, sometimes lawyers just get in the way.
No. Is there any impetus on the Hill to adjust HIPAA to facilitate
No, or at least
sharing
not,
of de-identified data?
So not that I'm aware of, but there would be incredible power in being able to do that, and we have tried all sorts of models, and it's just hard. It's hard in real life.
Web comment.
Had a special viewer, McLean Sunderland, who happens to be the son of the presenter, and he said he wanted to congratulate you for being able to stay up so far past your bedtime.
It's a red microphone somewhere. Oh no, blue microphone.
Hi, Will Angel. I'm a member. On the clinical studies side, what are some of the regulatory things that could be done to help accelerate those trials?
Well, no, I'm laughing because that was the topic for the meeting I was at yesterday. Literally, and it was attended by pharma and by CROs and by academic. We all want to do this. The question is, how can we efficiently do this? And it is particularly challenging in this space because it's so new. And a lot of the things we have to do in these clinical trials that these pharma companies are asking us to do, radioactivity samples and things like, it's outside of their sweet spot, and they don't know quite how to do it. And then the CROs, the contract research organizations who are supposed to be advising them, sometimes don't know what they're talking about either. And then things just get terribly inefficient over time. And so we were trying to literally identify those efficiencies, try to create databases of sites, trying to pre-qualify the quantitation of the equipment such that that doesn't have to be done with every trial. Because if I did it last month, why do I have to do it again with a different company? So we're working on it. But it's a huge problem. It's a huge problem.
Well, thank you for working on it. My mother is currently undergoing radiopharmacological treatment for cancer. So thank you.
Okay, well, best of luck to you and her.
I have a question. Actually, it's to ask you to make a comment about I think you were saying something about the universal scaffold of solid tumors that you have.
Yeah. So the fibroblasts. Yes. So that was the FAPI up and coming relatively general tracer. So these FAPI compounds bind to these cancer-specific fibroblasts that are almost always form around cancer cells, and they have chemical entities on their cell surface that these inhibitors actually bind to. Actually quite robustly. They are trying hard not just to use these diagnostically, but also to do therapeutics. So there's some therapies associated with them. We're involved in some clinical trials there. The uptake isn't quite as high with those FAPI compounds as they are like with the prostate and the others, so we can't get quite as much activity there. So it might be a little bit more of a challenge. But it's a fascinating concept to be targeting the tumor microenvironment rather than the tumor itself. But it's the same receptor-based binding that we're talking about, just different target.
And I realize this is already challenging enough, but is anybody doing combinations so you can have two different radioisotopes and two different binding agents, but they're both targeting the same cancer cells, so you have a better profile of the radiation exposure?
So I think the answer is not yet that I am aware of, but I would also say this is really young.
Yeah.
I mean, the first one was approved in 2018. And the number of ideas that are out there are numerous. But combination therapies in theory are the right things. Combination, because it's near term. Combination radionuclide-based ones, not yet, because we don't, I think, have that many approved agents to do it with.
Well, if there are no more questions, there is a question. Blue microphone. Name, member, question.
Sure. I'm a PSW member, a new member. I was wondering, you mentioned neuroendocrine tumors and them being a little specific, but I was wondering, as far as treatment, is it the aim to apply this or irradiate them so that they disappear, or is it more to control them, get them to shrink, and then just extend the life of the patient? And then also, are there any other therapies that may be combined with this, or that this could be a vehicle for that you envision?
Okay, so good question and right question. So I really don't mean to imply in any way that any of these are cures. You can make those cancers go away, but then almost always they come back. But sometimes you buy 3 months, sometimes you buy years. I could have brought up, and I'm not going to show right now, but the trial that got the Lutathera, which is the somatostatin receptor therapy for neuroendocrine tumors, had progression-free survival as its endpoint, which means the cancer isn't getting any worse. And they had 60% of the patients after 3 years had progression-free survival. Now, neuroendocrine tumor it's an indolent tumor. It's very slow growing. People live for a very long time with it. So as long as you can keep it at bay, you're doing a great job. And so it does this. Sometimes you get dramatic reduction in tumor bulk. Sometimes not. Sometimes it almost completely goes away. But trying to keep the cancer at bay is almost always the goal, because you can almost never fully eradicate it. And I'm just going to say one more thing, because these trials in general, they only occur in patients when they kind of have later stage cancer, when it's kind of gotten big and out of control. And so at that point, you're just trying to control. But it does make you wonder if we could go back and treat these early on when there's not that much disease. Can you actually cure them? those clinical trials are really hard to do. That's why they do the late stage ones first, because you can get good data, and they gradually march back to earlier and earlier stages of disease. That's what's happening in prostate cancer. The therapy is being used in earlier stages, but ideally, maybe you could get it where you can attack it to the point where you can actually kill all the cancer before it gets too far out of hand.
No, you gotta use the microphone.
How long do you think it will take to get to that point? So you're now treating, advanced cancer. So when can we see like the earlier interventions then?
I think that's too hard a question to answer, and it's pretty far into the future, largely because these endpoints are really hard. So survival endpoints, I mean, that's why they like people to do late stage cancer things, because you can say, well, they normally live 6 months, we got them living 12 months, so the clinical trial doesn't take that long. If you catch cancer early and people are living 5 or 10 years, how long does that trial need to be in order to show efficacy? So it's a challenge.
So I'll ask one other question. When you look at a cancer that's metastasized, different sites have started at different times. So can you say that presumably they're an analog to the different stages from initiation to, or are the cells different after they metastasize?
So this is a really good point, right? So I said 95% of prostate cancers, for example, express this PSMA, right? So, but when you get to late stage cancer, because as they're dividing, they're mutating, right? And they can differentiate to the point where sometimes they won't express the PSMA anymore. And then you can't use that treatment anymore. And in fact, if there are a few cancer cells that don't have the PSMA on it, but you're killing all the other ones, the ones that are going to divide are the ones that don't express PSMA abundantly. And so you're enriching those populations, which makes it challenging, which is why they're trying to do the combination therapies, so they can hit it from a couple different angles.
Is there no more questions? Then let's thank we got a question here. Last question. If we can get a microphone over here in the third row, a red microphone. Name?
Emily, not a member.
Okay.
And I was going to ask, what's the biggest barrier to manufacturing these expensive machines? Besides costs,
So it depends. Are you talking on the imaging side, or you're talking on the production side?
The production.
Okay. So facility production is really expensive because of the need to create good manufacturing practice facilities. So like clean rooms, things like that. It's really expensive to have to do that. The hot cell, we're looking at replacing some hot cells, and replacing a couple of hot cells is $1 million just for the hot cells themselves, because they're HEPA filtered as well, because you have to keep the air clean in them and stuff like that. So it's not just a bunch of lead or anything like that. So the setup cost of these manufacturing sites is really high. And it is crazy, because you think about a couple of hot cells, $1 million. I can buy a cyclotron for a couple million. But then you have to put it in a concrete bunker that has, and you start pouring concrete that's 4 feet thick. And then that ends up being, expensive just in and of itself. So the setup ends up being the big expense in that right now.
Any other questions? If not, we're going to thank our speaker now, and then I'm going to give him some gifts. So before you go, we have just a few gifts. One is a framed announcement of your talk. Another is our rosette with 3 stripes on the side, which is evocative of the symbol for inductance, which was studied by the founder of PSW, Joseph Henry. And the third is a signed copy of Volume 17, first edition of the PSW Science Bulletin, founded as the Bulletin of the Philosophical Society of Washington. This is a compendium volume, and you will find the formative papers. The first address of a president, who was Joseph Henry, and he lays out some still useful pointers about how scientific discourse should be conducted and what these organizations are for. You'll also find a list of all our Nobel Prize winning speakers and all our heavy speakers, and a number of other interesting things that will help you on your airplane flights. And with that, thank you so much.
Thank you.
A few housekeeping announcements.
This was fun.
Before you go, just a few housekeeping announcements. The next meeting will be the 2,525th meeting on November 21st. The speaker will be Jeffrey Alexander, who is the Director of Innovation Policy at RTI International. He'll be speaking on the real returns on public research investments. And there are real returns, for those of you who had some doubt. Other upcoming lectures posted to the website include Rosalie Lopez from JPL speaking about volcanism and cryovolcanism throughout the solar system. Davey Smith from UCSD, who will be talking about carrying out rapid autopsies, by which she means autopsying deceased very quickly after they die in order to understand, as he likes to put it, where HIV hides. Then on January 9th, opening up the new year, we'll have Bernard Carlson of the University of Galway and of UVA, who has written a book about Nikola Tesla, and we'll be talking about him, not the car company. I may talk about the car company for all I know, but the main point is the annual President's Lecture will be on January 23rd. So if you like black tie affairs, mark your calendar. Otherwise, you can come to the lecture as usual, and the speaker will be Gordon Guyette of McMaster University. He's going to be talking about GRADE, a formalized system for rating the quality of evidence that informs healthcare. So it's basically a clinical study rating system. Additional speakers will be posted to the website. There are a number, but I'm going to keep them secret, so you'll all be guessing. If you have suggestions about speakers, you can email them to me. I have lots of them, but it's always good to have more. And with that, let's thank our crew for their work tonight. And with that, we'll take a motion to adjourn tonight's meeting to the social hour. A second. All members in favor? All members opposed? Meeting is adjourned to the social hour. The beverages and the refreshments at the back of the room.